Thanks for having me. This is I'm very excited to be here today and to give this talk. I'll go a little bit into my background. I am an archaeologist. I've been working with the Reich Lab for about 5 years now. And it's really a unique time in ancient DNA. The growth of the field, the sort of rapid expansion of it happened when I started in the Reich Lab. So I'm a bit of an outsider on the genetics perspective of things. But coming in from an archaeological background, this technology that we have now to examine the people who live in the past is very exciting, and it is revolutionizing the field. So maybe the first thing I want to do for starting out is just to make a note on my title slide, and that really sort of speaks to my archaeology background. You can see I've made a note about the presentation containing images of skeletal material. This is something, denoting this as something that's actually pretty standard practice in archaeology, especially archaeology in North America. There's a lot of sensitivities, cultural sensitivities, about showing the skeletal remains of Native American ancestors. And it's important, I think, to make a note of that before a presentation occurs. And in my experience, one of the interesting things, this shows sort of the different ethical approaches between the two fields. This isn't something that's maybe common practice in the medical field or the genetics field. But again, I don't think that one way is right or wrong, but it shows sort of this dichotomy between the two fields and some of these issues that we have to navigate as archaeology becomes more integrated with DNA or ancient DNA. So ancient DNA really has had a rapid and significant impact on archaeology. My background, as I said, is in archaeology, and as Sarah mentioned in my introduction, while I was working on my dissertation and doing graduate school, ancient DNA was kind of in the background. I had heard about it, but it was never something that I had ever thought that I'd be able to integrate into my own field work. When we learned about ancient DNA, it was usually well-known cases like Denisovans, identification of Denisovans, or the publication of the Neanderthal genome. There was some other well-known studies that I'd come across occasionally, but it really wasn't part of everyday archaeological practice. I think in the last 5 years that has changed. Ancient DNA has really begun to revolutionize archaeology, and because of that, there's been a sort of an explosion of discussions about ancient DNA and how it's impacting the field. So there's been entire journal issues dedicated to ancient DNA, in leading archaeological publications. There has been multiple conferences that focus on ancient DNA, the ancient DNA revolution in archaeology, and the ethical implications. There's now an NSF-funded ancient DNA ethics project that's studying ethical issues of ancient DNA, particularly in North America. And the American Society of Human Genetics has even published a piece in August, and I'm going to talk about this piece as I go through my talk, about ancient DNA and ethical guidelines that they proposed and that the ASHG has signed off on. They've sort of approved these officially when the article was published. So my talk today is going to be organized into three broad sections, although I'm going to spend most of the time in the first, which is what are the key ethics issues in ancient DNA research. I'm going to, towards the end, talk a little bit about why there has been so much focus on ethics in ancient DNA and what I think are steps to move forward. And I forgot to mention too, I'm very open to questions. So if anyone has a question, I can't see it because I'm in presenter view, but if you do, please raise your hand or post in the Q&A and the question will get passed on to me. I'm happy to answer any questions. Okay, so this first point, like I said, the main part of my talk is going to focus on the key ethics issues in ancient DNA research, and I've divided this into 5 subcategories. So what I think these key issues are, if you had to group them together, are questions about scientific best practices and open data, engagement with indigenous communities, sampling best practices, archaeological collaboration and interpretation. I'm not going to focus too much on that here. If I'm giving a talk to a more archaeological audience, I might spend more time on it, but I think some of this isn't really pertinent to what I'm going to be talking about today. And then modern political implications, and this is broad. This applies both to modern genetics and ancient DNA. for the first 2 points, and really throughout the talk, I think case studies really can help illustrate some of these issues best. So I'm going to spend some time talking about some well-known case studies to highlight these ethical issues and concerns that people have raised. And I think there's no better than the Kennewick Man or the Ancient One. So some of you might be, it's a very well-known archaeological study outside of ancient DNA. Ancient DNA is actually sort of the end of the Kennewick saga. But even the name, this sort of goes back to my title slide. It speaks to some of these ethical issues where, the archaeological name assigned to this set of skeletal remains is Kennewick Man. But indigenous people, the descendants, people who feel they're descendants of this individual, call him the Ancient One. So I'll switch back and forth in the talk. Indigenous groups, would probably strongly prefer that the Ancient One is used. So I'm going to try to use that. But, my archaeological background, when I was learning about this case years ago, Kennewick Man was still sort of the standard archaeological nomenclature used. So again, I'll go back and forth a little bit. They mean the same thing, referring to the same set of skeletal remains. So some background. In 1996, there were some skeletal remains that were found on the banks of the Columbia River in Washington State as the Army Corps of Engineers was doing some work there. At the time, the Corps of Engineers sort of immediately wanted to repatriate these remains to the local Colville tribe. So there's a law in the United States called NAGPRA. It stands for the Native American Grave Protection and Repatriation Act. It was passed in 1990. And basically what it says is that any Native group that can demonstrate cultural affiliation to skeletal remains found in federally funded museum collections or repositories need to be repatriated to those groups. Now, there's a lot of aspects of the law that I'm not going to get into here that often complicate things. But what happened with Kennewick was that the Army Corps of Engineers wanted to immediately repatriate the remains to the Colville tribe, but the initial morphological assessment of the skeleton showed evidence that the skeletal remains weren't Native American. In fact, an early facial reconstruction you can see here made Kennewick look like a very well-known actor on Star Trek Next Generation, compared to a Native American ancestry, or so what you might expect a Native American person to look like. And in fact, the anthropologist who did the initial assessment thought that the skeletal remains were that of a European settler dating to the 17th or the 18th century because of features that he felt were, and he used the word Caucasian. But it became clear pretty soon after this initial assessment that if these were Caucasian or European remains, they were very old. There was a projectile point, an arrowhead that was found embedded in the pelvis of the Ancient One that was a very archaic style, very old. And then radiocarbon dating put the skeletal remains at around 8,000 BP. So this is one of the most ancient individuals, complete skeletons that have been found in North America. And these two combined things, the morphological features indicating, that the Ancient One was maybe Caucasian or had more of European ancestry, and then the ancient date set off a firestorm. Kennewick was covered widely by the press. Excuse me. And what happened was white nationalists, far-right activists picked up on the story and said, look, there was a European here, there was a white person here 8,000 years ago. This proves sort of that white people have justifiable land claims in the US, and the displacement and genocide that happened to Native people is justified because there was white people here 8,000 years ago. That was one of the reasons why this got picked up by the press. Another reason is it has to deal with these NAGPRA issues. So despite the fact that anthropologists, early anthropological assessments classified the skeletal remains as Caucasian, Native people still felt that Kennewick should be repatriated. But because of this ambiguous background, the Ancient was initially classified as culturally unaffiliated, meaning that archaeologists couldn't tie him to a specific modern native group. So it basically led to 20 years of lawsuits and back and forth between the Smithsonian Museum and archaeologists and native groups about who ultimately would have final possession of Kennewick. What ended up happening was in 2010, an addendum was passed for NAGPRA that said that culturally unaffiliated remains can also be repatriated to tribal groups. So the Ancient One, after that was passed, basically was going to be repatriated to the Colville tribe. But before that happened, there was an ancient DNA study that was conducted, led by Morten Rasmussen and S.K. Willerslev. And this study, it sought to do a few things. I'm going to go into the study a little bit. And it was actually, ultimately, the study was very well received for what it found. So what the researchers did was now what seems to be some basic ancient DNA analysis. The study was published in 2015, but they used F3 statistics to admixture and PCA to look at the ancestry of Kennewick. And what they found was that the Ancient One, as should be expected, had ancestry that had much more affinity to Native American populations than European populations. So if you look at panel B on the figure here from Rasmussen et al. 2015, you can see the F3 statistics where the Ancient One has much more affinity with modern Native American populations than other populations. So Central European is the blue square, and then East Asian populations. There's more affinity to East Asian populations, which is what we would expect. They also looked at the direct ancestry of Kennewick. They tried to find who might be the direct descendants of Kennewick. And so what we see on the panel here are more F3 statistics. We have Kennewick in panel A, Anzac I, which is another very ancient individual from North America, associated with the Clovis culture. It's individuals from Montana, and actually is older than Kennewick, about 10,000 years old. Then we have the modern Colville people, and then modern Chippewayan people. And you can see panels A, B, and C are all pretty similar, and this is what we would expect. So there's been archaeological evidence and genetic studies that have looked at population demography and history of Native American populations. And essentially there's been multiple waves, but the two primary waves are what have been called Ancestral A and Ancestral B. Ancestral A are tied to the initial peopling of the Americas. So these are people that, initially, or for a long time, archaeologists thought came over across the Bering Land Bridge. But now there's pretty strong evidence that people came prior to the ice sheet opening up and allowing them to come down into the continents, and instead followed the coast, the Pacific coast. And that's what we're seeing with A, B, and C, is basically this Ancestral A ancestry. Ancestral B are Athapaskans or Na-Dene groups. They came later into North America. And so what this is essentially showing is that Kennewick's ancestry is, more closely associated with that Ancestral A population than Ancestral B. The researchers also did a direct ancestry test. And if you look, they compared the Ancient One's DNA to 2 modern Colville individuals, 2 Athabaskan individuals, and then 2 Caritiana individuals. And so ultimately, what the paper did was put the final nail in the coffin for this question about whether or not Kennewick is Native American. He was. There's no question about it based on the genetics. Now, with that being said, there have been some questions raised about the science of this paper. So first of all, as I said, this was done in 2015, and there was no ancient DNA comparison made. That's partly because there was really no ancient DNA samples from the Americas at the time. There's some now, there's not many more. So to be fair, the authors didn't really have much to work with in terms of ancient DNA. But the modern groups that they choose, especially with the direct ancestry that they chose, especially with the direct ancestry test, is a little questionable. I'm going to flip back to that slide if I can, just to look at this again. Like I said, when they did the direct ancestry test, they looked at 2 Colville individuals, 2 Athabascan individuals, and 2 Caritiana individuals. These are very distinct groups. Colville are the most geographically proximate to Kennewick. Athabascans are somewhat geographically approximate, but they're part of that later ancestral B group that I mentioned. And then Caritiana are an Amazonian group, so much further south. And while their data shows that the ancient one was, had direct ancestry that's closest to the Colville, there really wasn't much or any testing done of other modern groups. It would be interesting to do the same test on, say, modern Hopi or other groups from North America that have that ancestral A lineage. And my suspicion is that they would have probably pretty equal or close to the same probability for having direct ancestry as the Colville group does. But this was something that the authors just didn't address. The other thing, and this is going to play into my talk as I go, is data access. The data that the author used, the Kennewick genome is fully public, but the comparative data from these modern populations is not, and it's been difficult for researchers to access that and do replicability studies. So that's something that's come in question too, at least from the scientific end and scientific ethics. But like I said, overall, the study has been very well received publicly and by archaeologists. It's used as an example of how ancient DNA can be used ethically to help resolve issues dealing with repatriation and indigenous sovereignty. So it's a case study that sort of shows how some of these different issues can be addressed and how ancient DNA can be used to help with archaeological issues. The next case study I'm going to talk about comes from Chaco Canyon, which is in the same region where I do my archaeological research. But it's the inverse of what I was just talking about. Here, the science is unquestioned. It's very strong, and there really hasn't, at least as far as I'm aware, been any questions about the science that was done for the project, but it almost immediately was questioned on ethical grounds. So this study was published in 2017. A little bit of background: Chaco Canyon is located in north central New Mexico. It's a very well-known national park, and it's well known because of the architecture that was there. This is the largest standing architecture, masonry architecture in North America prior to Europeans arriving. So Chaco Canyon dates to about 1,000 years ago, and it was sort of the center of what's called the Chacoan phenomenon or the Chaco culture. And archaeologists have been debating for over a century about sort of the details of that culture, whether it was hierarchical, whether there were elites or not, whether it was egalitarian. And so ancient DNA was used as a new line of evidence to explore this. And what the researchers did was examine individuals from room 33, which is in the top right panel, and the burials there were very rich. There's a lot of grave goods that were found with them that suggested that they were elites. So the researchers did an ancient DNA study combined with radiocarbon dating, and what they found was that these individuals were closely related matrilineally. And what they argued was that this is evidence of a strong matriline, a dynasty, a matrilineal dynasty in Chaco Canyon. It really helped resolve a longstanding archaeological question. But as I mentioned, almost immediately there was strong pushback on the article from an ethical perspective. So again, like Kennewick, there's a bunch of background that I don't want to get too into the weeds on, but it deals with NAGPRA again. So the individuals at Chaco Canyon that were used in the study were excavated decades ago, early 1900s. And they've been stored at the American Museum of Natural History in New York since. They were classified as culturally unaffiliated. I mentioned that term earlier with Kennewick and NAGPRA, even though a lot of archaeologists and modern groups in the Southwest, modern indigenous groups in the Southwest, feel that they shouldn't be. They feel that there's a clear link between Chaco Canyon and modern Pueblo groups like Hopi, Zuni, Acaba, Laguna. So because of this, many people feel that there should have been strong indigenous consultation, work with native groups prior to the study being done or even initiated. Researchers didn't have to do that because the remains are classified as culturally unaffiliated, meaning that after submitting a proposal and making all of the proper requests, you can access those remains to do studies, radiocarbon dating, ancient DNA, isotopic studies. That wasn't done in this case. And now in the Southwest, any work that's done on human skeletal remains almost always has to go through some sort of engagement or consultation with Native groups. If remains are culturally affiliated, the groups have to give approval before any work can be done. If they're culturally unaffiliated, as I mentioned, there doesn't have to be an engagement or consultation, although it's very highly recommended, especially in cases like Chaco Canyon. So a lot of the pushback was because these remains were studied in the first place. Native groups are, they feel that their ancestors have been improperly stored in museums for decades or even centuries. They want them back, and any further destructive analyses that occurs on those remains is an affront to modern indigenous groups. So this study has really sparked sort of an ethical tidal wave in ancient DNA, especially in North America, relating to North America. So this is that article that I mentioned earlier that came out this year that was published by the ASHG as guidance. So what the authors here did was come up with 5 recommendations for paleogenomics for proceeding in an ethical manner. I'm going to highlight these now and come back to them a little bit later. I'll note too that many of the authors on the paper are indigenous themselves. They're Native scholars that are working in ancient DNA or genetics. But what they recommend is that researchers formally consult with communities, and this includes, prior to any study occurring, getting approval from a group for the study to occur. Addressing cultural and ethical considerations. So what this means is that while the study is ongoing, researchers have a dialogue with indigenous groups and are reporting data back to them and early versions of the manuscript, so that groups can have feedback. And if at any point the local community or the indigenous group becomes uncomfortable, the guidelines say that if the community disapproves of the project and decides not to proceed, then ASHG believes the researcher should respect the community stance and the research should not be conducted. So basically what this means is that local groups have veto power over a study once it's been initiated if they're uncomfortable with the study. They say that researchers should engage the community and support capacity building when possible. This includes things like education and training, setting up advisory groups, giving co-authorships on papers, and having constant communication updates. They urge ancient DNA research to develop plans to manage data and report results. This could include holding back data if it's too sensitive, if different groups feel it's too sensitive to make public. And then develop plans for long-term responsibility and stewardship of both the data and relationships that researchers have forged with indigenous groups. So I'm going to move on now and talk about some of these other ethical issues that have come up. Another, I would say maybe the next most significant issue is sampling best practices. And really what this stems from is the fact that ancient DNA is destructive. We have to grind up some bone element to get powder. And then really, in the last half decade, there's been a focus on the petrous bone. So the inner ear bone, the cochlea, which is surrounded by the petrous portion in the temporal bone, has been proven to generate many-fold more ancient DNA data than any other skeletal element. So there's been really a focus on using these skeletal elements, but the thing is, these are morphologically important, and there's only 2. It's not like a rib or a tooth where there's multiple elements, or even bones that like femurs where there's 2, but there's much more material available. So there's a concern about the destructive analyses that are being done on the petrous, even though only 37 milligrams of powder is needed to generate ancient DNA data. So, some of the issues surrounding what's been called sample hoarding or processing. There's a few ancient DNA labs, including the Reich lab, that process the vast majority of ancient DNA samples. So archaeologists are concerned that samples are being filtered into these few labs, that have control of most of the world's ancient DNA data and samples. There's a concern about this analysis, the destructive analysis, as I've already talked about, labs hoarding samples. Another key part of this is perpetuating what is considered to be a colonialist legacy or parachute researchers. Basically, a lot of these skeletal remains were collected in the late 1800s, early 1900s, and against the wishes of native groups in the Americas and Africa and Australia. And then also, these labs have been accused of following a sample first, ask question later's approach, meaning collect all the samples that you can, and then decide what you want your hypotheses to be or what questions you want to address. I'll just make a note here that this certainly isn't our approach in the Reich lab, but there is a sort of a feeling amongst archaeologists that this is happening. There's been some proposed solutions. I'm not going to go over each of these in detail because I'm going to have more solutions at the end. But I just want to point out that this is something that we take seriously, and we've been thinking about and trying to come up with ways that we can, mitigate the destructive analyses that we're doing and address some of the concerns that archaeologists have. Excuse me. So moving on, like I said, I'm not going to spend too much time on this point because it is more archaeologically focused. If I had to sum it up, what I would say is that what this deals with is archaeologists getting mad about big arrows on maps. So essentially what's happened is in the last 20 or 30 years, archaeologists have moved away from big picture population questions to more individual focused questions about particular cultures or even particular individuals. And then, as ancient DNA has come into the field and really started to address questions about population history or demography, some archaeologists have been concerned that, archaeology is losing some of its granularity. And so essentially what's happening is that this is more of a theoretical debate about how ancient DNA data should be interpreted and used, with genetic studies being conducted in one way and then archaeological studies being conducted in another. Like I said, I'm not going to spend too much time on this for now, but a lot of it has to deal with, these issues of migration and population movement and things like that. Something I do want to spend some more time on is the modern political implications for ancient DNA studies. So I'm going to start with an example, another case study from Oceania. This is from an issue of archaeology in Oceania, where there's been a few key studies that have come out in the last years that have really overturned archaeological understanding of the ancient populations there. And so there's been a dialogue between archaeologists and geneticists about some of the results. Essentially what happened is that prior to ancient DNA studies, many archaeologists had argued that the Lapita culture, which is sort of the first to appear in Oceania, really sort of spread throughout Oceania, and that the modern day people that live in Oceania are descendants of that Lapitian culture. I generally don't like to put a lot of small text up on a slide, and I'm not expecting you all to read it. And I generally don't like to read it myself, but I think that I'm going to do it here just because I think there's no better way to illustrate this point. Anyways, that was the archaeological understanding. And then ancient DNA evidence has kind of turned that on its head. So I'm going to pick up sort of about a third of the way down in the first panel. Especially the results could potentially be used for future studies without the consent of the participants, as in the case in Post et al. This decision was in part influenced by the recent appearance on web forums of old-fashioned colonial theory that the Kanaks were not the first settlers, relying on publications like the two discussed in the present forum. By declaiming, even if it's not their goal, on slim scientific data with binary conclusions, the historical rights of the indigenous populations in southern Melanesia, Western geneticists perpetuate a form of intellectual colonial power and do not contribute to highlighting what is the prevalent characteristic of the deep past of Southwestern Pacific, its extraordinary complexity. And then the next panel, this is from a different article within this journal issue. Finally, I am uneasy with the implications of these studies for descendant communities across remote Oceania. Archaeologists have long validated their Lapita ancestry. We have worked closely with community members, and we have empowered them with our assurances that they had come first. In Vanuatu, public promotion of this ancestry has been extraordinary through media, through training workshops, in cultural center exhibitions, and in 2005 in a Lapita stamp series with Lapita peoples intentionally cast in Ni Vanuatu likeness. In New Caledonia in 2000, we called home the Lapita peoples of near and remote Oceania to celebrate the 50th anniversary of the first Lapita site excavations and to witness the reintegration of Lapita tribes at a traditional Kanak ceremony at Kane. One can only feel a collective sense of betrayal in all of this, regardless of the scientific significance and the insights that Post et al. And Lipson et al. Have generated. So basically what this is saying is that archaeologists have been telling Lapita people, telling modern day people in Oceania one thing about their ancestry, that it's Lapitian. This has become actually integrated into their culture. It's part of their cultural identity and who they understand themselves to be. And now there's a new class of data coming that's totally upending that. And so there's concern about the harmful impacts that might have on the identity people feel they have in Oceania. I'm going to move on. Here's an extreme example that some of you may have even come across of ancient DNA and its misuse. So I'll say in the previous example, the data weren't misused, but there's a concern about how it might impact how people perceive themselves. In this case, this is an example of ancient DNA data being misconstrued and used inappropriately. So essentially what happened is there's a study published in July 2019 on skeletons from the site from the city of Ashkelon. The study demonstrated that the Philistine population in Ashkelon had ancestry most similar to contemporaneous populations in Southern Europe. This led the researchers to conclude that the Philistinian population had recently migrated to Ashkelon and the Middle East. After the study was published and picked up by the media, Benjamin Netanyahu, the prime minister of Israel, sent out a series of tweets that misconstrued the results. Essentially what he said, or what he did, was he used the study to claim that Philistines and therefore modern day Palestinians have a recent connection to land in Israel, and that Jewish populations there have been there much longer. And so he's using ancient DNA data to make claims about land rights. And you can see where this leads, about justifying annexation of lands, more lands in Israel, and sort of throwing out Palestinian populations. So this was it was a very controversial tweet that was sent out, and there was a lot of discussion by the researchers on what to do. Ultimately, they decided not to engage with the tweets because they didn't want to highlight this more, but it shows how ancient DNA data can be misused, and really how there's a growing concern both in ancient DNA and modern DNA about how this data can be misused by white supremacists and nationalists and xenophobic people, taking data and just totally misconstruing it. I'm going to leave that there for now because I'm running short of time and I want to cover some of these other points. So as I said, one of the things that I wanted to talk about is why there's been so much focus on ethics in ancient DNA. So there's all the reasons that I've just spent time talking about. But another key reason I think is sort of the impact that ancient DNA is having on the field as a scientific revolution. So archaeology has gone through a number of revolutions. Perhaps the most well-known one is radiocarbon dating, where essentially prior to radiocarbon dating, archaeologists could only do dating through relative chronologies, ordering things by knowing whether, figuring out whether one thing came before the next. Then radiocarbon dating came along and totally upended that. And there was all sorts of debates about radiocarbon dating. In fact, archaeologists weren't really receptive of it at first. It's similar to what's happening in ancient DNA now. It also stems from these two different fields, which have different ethical backgrounds and different ethical approaches coming together for the first time and trying to navigate a way to merge the fields together. And then I think another reason why there's been such attention on these ethical issues is just DNA is in the ether. these two curves are very interesting to me. The one on top, it's from January 2019, so it's old now, but it shows the growth in consumer genetics companies, things like 23andMe and Ancestry and at-home ancestry testing, and the growth of ancient DNA. And you can see the inflection point is almost identical for both. So, people are talking about DNA and ancestry and genetics and all these things more. And because of that, and the ethical issues that have been raised about them outside of ancient DNA, some of that's permeating into ancient DNA where it otherwise might not have. So then how to move forward? As I've mentioned, there's been a lot of debates and discussions about ethics in DNA, and I think there's some really positive steps for how we can address some of these issues. Clearly, there's a need in places like the Americas and Australia and Africa to work with local communities. There's questions about the degree to which this needs to happen and the degree to which non-researchers, non-scientists should have a say in how research is conducted and data are published. But despite that, it's clear that we as ancient DNA practitioners need to work with the groups who feel that, their ancestors are being studied. And there's been some really good case studies. This is one I wanted to highlight from sub-Saharan Africa, where the archaeologists that we collaborate with have really strong relationships with indigenous groups there. And throughout the process, we're communicating with them and sharing results. Another sort of newer way that ancient DNA might be used to address ethical concerns is through what's been called restorative justice. So, I've talked about repatriation a little bit, but basically in this paper that was published last year, what happened was we examined a set of remains that were in the Americas for a paper that dealt with ancient Peru. And there's a question on whether their provenience, whether or not they were actually Peruvian. Our ancient DNA study proved that. And after the data demonstrated this, the remains were repatriated back to Peru. And it was sort of viewed as a mutually beneficial, positive thing that happened. And so myself and another postdoc in the lab, Kendra Sirach, are actually right now trying to put together a project that examines whether or not ancient DNA can be used as a tool to help in repatriation and sort of right some of the wrongs that indigenous groups feel have happened to their ancestors being excavated from the ground against their will. I wanted to point out this map too. This is a map of every published ancient DNA sample as of a month or two ago. And there's a clear discrepancy here. Most of the ancient DNA work that's been done has happened in Western Europe. You look at places like North America, Australia, there's no samples that have been published. This is for a variety of reasons, some of which I've talked about. In some, there's just a reluctance by Native groups to allow researchers to work with the skeletal remains of their ancestors. But there's also research bias by researchers. They worked in places where it's easier to acquire samples maybe, or where there's big labs. And so I think that one of the things that ancient DNA needs to do in the future is to work to correct this. I think there is a way forward to generating ancient DNA from places like North America that respects modern indigenous groups while also adhering to scientific best practices so that we can learn about the ancestry and the history of people in these regions like we have elsewhere. I wanted to touch back on this ASHG article and the recommendations that were made there. So I think that this is a very good starting point for an ethics discussion, especially in North America. But as I've mentioned, there have been some concerns raised about whether this crosses a line and starts to violate scientific principles, I guess, especially principles in terms of having veto power over a project once it's begun and not reporting results that might be considered controversial. And then also data management. There's been a strong push in ancient DNA by some for data to not be made public and be put in restricted databases, which is not something that I personally feel comfortable with. One of the things that I've really enjoyed about working in ancient DNA is how the data has been so open and accessible. That's typically not the way things have worked in archaeology in the past. Archaeologists like to hold on to their data and not share it. So it's really been nice for me to work in a field where data is open and testable. So, again, these are starting points, I think, in the Wagner paper that some of us are coming up with a response to, which is what I'm going to close on, where there was a meeting in November between a group of ancient DNA practitioners, archaeologists, museum curators, indigenous people that try to find what we call common denominator principles that we all as stakeholders can adhere to, that allow us to do scientific studies, but also, address the concerns, some of these ethical concerns that have been raised. So, the first point is that researchers must abide by all regulations in the places in which they work and from which their samples were drawn. So one of the key takeaways from the Wagner et al. Piece, the ASHG piece, was that it really was built on a North American model and a NAGPRA model. That model isn't applicable in other world regions. For example, Mexico, we've got very strong collaboration with some Mexican archaeologists, and the way they feel about showing skeletal remains on slides, doing destructive analyses on skeletal remains is entirely different than in the United States. And so, what we propose is that, archaeologists and geneticists need to be aware of sort of what the local feeling is about working with skeletal remains and generating ancient DNA from them. We can't come up with stringent guidelines that are developed in one place and try to apply them globally. With that being said, we feel that researchers must prepare a detailed plan prior to beginning any study. Again, in my experience, this has been the case, working in the Reich lab, but there's clearly concern that, some researchers are just collecting samples before really having a good idea of how they want to use them. And that's something that we want to make sure doesn't happen. Researchers must engage with stakeholders to ensure respect and sensitivity to stakeholder perspectives. I've already mentioned, I think this is clear. There's debates about the degree to which this should occur, but at a minimum, I think that it's necessary to share a manuscript with local groups prior to publication so that they can give feedback. Again, this might not be appropriate in all places, but in places where there are local indigenous groups that have ties to the skeletal remains that we're working with, I think something like that is critical. Researchers must minimize damage to human remains. I've already touched on this point too. Like I said, we only need 37 milligrams of bone powder to generate an extract. That's really not that much, but when you're talking about skeletal elements like the petrous that are morphologically important, a little bit of destructive analysis goes a long way. And so we are constantly trying to find methods and refine our methods that minimize the impact that we have on skeletal remains. And then researchers must ensure that data are made public and available following publication. Again, this is something where I think that there is some tension between archaeologists and some indigenous groups and ancient DNA practitioners, but it's just my opinion. I think the opinion of a lot of researchers that having data made public is good science, and it allows for testing and replicability. And it also allows anyone with sufficient knowledge and training can look at these data and sort of see for themselves what they say and what the data might imply. I think having open data is only going to be a benefit moving forward. So I'm going to wrap things up. I'd like to thank the John Templeton Foundation, who is a major funder of the Reich Lab, and in particular, my position in the lab. Of course, David Reich, my PI, the Reich Lab technicians who have generated a lot of the data that I talked about, the ancient individuals whose ancient DNA we study, the indigenous groups whose ancestors we study, and then Diane and Sarah and Christine and everyone involved, Jim, who invited me to give a talk for the Primer series. It's really great to discuss this with an audience because these are things that we talk about internally a lot. And I think it's important to show that we are really giving a lot of consideration to these issues and the impact that they have both on the scientific results and the descendant communities whose ancestors we're studying. And yes, please let me know if there's any questions. Thank you.
Thank you so much, Jacob. That was really a wonderful and really interesting talk to listen to. We have one question posted at the moment that I will read. The question's from Anna Lewis, and the question is, are your common denominator principles being developed under a particular professional society or existing group? And how do you think they will be viewed, appealed to in comparison to the ASHG ones? That is a good question. So in full transparency, and we mentioned this in the article that we're working on, that the ASHG piece was really the impetus for this. We read the piece, and again, we thought that there was some good foundational principles that were established in that, but we also had some concerns about it. So we held a forum in November of over 60, maybe 70 practitioners of ancient DNA, whether that's archaeologists or geneticists or, like I said, indigenous communities, to really, our goal was to develop these guidelines so that they can be something that everyone can sign on to, not just applicable to North America. I'll say right now, we've been working on the manuscripts intensively, basically since December. And just this week, we're at a point where we're about to submit it. I don't know if I'm going to, at this point, say where we're going to submit it, because we don't ultimately know if that'll be the place where it comes out. But it is a high impact journal that we're planning to submit to. It's not going to be through an official organization. One of the things that we debated was whether or not the piece should be a response to the ASHG or something broader. We decided to go broader. I think, many of the people that are going to be co-authors on the paper would be happy to, if this was picked up by a professional organization. But our goal is, at least at this point, to have a response to the Wagner piece and give an example of what we think are common denominator principles that many people can subscribe to. Great. Thank you. There are no more posted questions, but I do have one about sort of what you would describe, and you've alluded to this a bit already, but as best practices for for researchers, genetics researchers who are interested in using existing samples that are currently in a museum and classified as culturally unaffiliated. It seems like there's a major discrepancy between the ethical awareness and understanding at the time that many of those samples were collected and classified and what would now be regarded as, the best practices for using those. And I wonder, in particular, for example, what are the steps for identifying groups that should be consulted at the very outset of the process?
That is a very good question, and something that we and me think about, I especially think about constantly. You're exactly right. There are a number of samples, in our lab and in other labs that were culturally unaffiliated, that have been processed, and labs are sort of sitting on the data because the ethics has changed so quickly since the samples were collected, that we really are trying to navigate and figure out what to do with them. It's complicated for so many reasons. One is that, it's sort of the ancient one case study is a good example. Sometimes the ancient individuals are so old that it's hard to connect them, at least in a sort of in a scientific or archaeological way, with modern populations. Demonstrating or proving, I don't know if that's the right word, that cultural affiliation is difficult to do for that reason. Another example might be, there's regions where multiple native groups live. My background's in the Southwest. That's what I'm most familiar with. And those groups don't always see eye to eye on things. And so you may have some groups that are, excited about ancient DNA and want a study to occur. And then on the other hand, you may have another group who is very anti-ancient DNA or genetics in general, even archaeology in general, and doesn't want a study to move forward. Yeah. And so the difficulty is, if both groups have equal weight or seemingly equal weight to, these remains as ancestors, what do we do? And it really is a struggle. I think, personally, we're doing injustice if we're just sitting on the data. At this point, it's the samples have been processed, and I think it's the ethically right thing to do to publish them. Maybe we have to do something like a mea culpa and say the ethical standards of the field now aren't what they were. We gathered these samples and they were collected, put in museums before the ethical standards are the way they are now. We're doing this to maybe help with repatriation of the skeletal remains. But we feel it would be inappropriate to have done destructive analysis on remains and now just to continue the data and the remains sitting in a researcher's lab. So it's a very difficult question. It's what honestly I spend a lot of my time working on, and there is no one right answer. Each case is almost a case study, and in terms of figuring out who to work with and who to engage with and what the best step forward will be. It's very complex.
Yeah, thank you. There's another posted question now from Rosie Husking. Community participatory research is often mentioned as an ethically beneficial approach. So are there outreach approaches by ASHG or others to increase recruitment of Native Americans into genetics research teams?
Absolutely. So there's a really good program called SING, which is the Summer Internship for Native Genomics, which is actively recruiting students to teach them genomics. Now, this isn't ancient DNA as a part of it. I think a main focus of SING is on more modern genetics, but there's been a lot of Native students who have been introduced to ancient DNA this way. We're constantly trying to come up with ways to do this too. One of the things that we're trying to do is actively recruit students for summer internships in the lab. And it's, like most things, COVID has kind of thrown a wrench in our plans. Part of the Templeton grant that I mentioned that David got was for outreach and engagement. And just prior to COVID happening, we had started bringing in archaeologists and scholars from different countries. I mentioned Mexico, to do things in the lab with us. But of course, that's all shut down now, and we're trying to figure out how to navigate things with COVID. But yeah, I think that's a very important point. And, it is something that's becoming more and more common on our end, in the Reich Lab. And what's really been successful for us is working with archaeologists who have community, deep ties to communities already. I mentioned the paper from sub-Saharan Africa that was really great for us because the archaeologists there had these really deep ties and connections to communities, and have done a lot of things to do capacity building there. So we're always trying to look to partner with archaeologists that can help us in those aspects of engagement.